So very good morning, my dear students. So in this planned phytopathology course, so today's sections we are going to discuss the definitions and terms related to the phytopathology, as well as the mode of attack of plant pathogens, right? Mode of attack of the plant pathogens. So first we will go through the definitions, terms and definitions, right? So because if you are understanding very clearly these terms and definitions, and it will be very helpful to you to answer the many questions, as well as if any question asked in anywhere of the career, then you can able to interpret and you can easily answer for that questions, right? So first term is the parasite. So parasite is the and living organism. Definitely it will be a different living organism. It may be the microbe, so it may be the high animal. So there's a broad term, right? It's a parasite. It's an organism living upon in another living organism. So there is a close relationships between these two organisms. So one organism get benefit, another organism will get detrimental effects. Therefore, we call it as a parasite. So this is one organism depend on other for the food as well as for habitat sometimes, right? This may be the higher plants, so higher parasitic plants. You would have heard about that Loranthus kescuta, or any microorganisms that are causing the disease. Commonly, we call it as a parasite, right? Because they are living to draw the nutrition. And pathogen. So pathogen is something different. This is microorganisms, not all the organisms. Bit we are narrowing down, right? So the microorganism that can cause the disease on host, any host, we call it as a pathogen. If it is causing on the plant, we call it as a phytopathogen, right? So pathogen is a microorganism that can able to cause disease on any host. We call it as a pathogen. It's a broad term. If it is causing on the plant, we call it as a phytopathogen. Right? So biotroph. So biotroph means if an organism require living host, we call it as a biotroph. If the plant or any host is dead, this organism can't use that host. That is, we call it as a biotroph. Right? For example, the nematodes, especially the Melidogyne, root node nematode. So they need biotroph. Otherwise, they will not. For example, if downy mildew, you cannot culture it in the lab, downy mildew and powdery mildew, because they need a living host. That's what you could not be able to culture it in the laboratory conditions. That is, we call it as a biotroph. Hemibiotroph means some of these plant pathogenic fungi initially require the living host. Then these fungus can kill the host. Then they will survive the dead host. That is, we call it as a hemibiotroph. So half of the plant pathogenic microorganisms are under this group, hemibiotroph, because they can initiate the infections if the host is living. Definitely, they will kill the host by producing certain enzymes. So we will discuss this one in the next chapter. And after the death of if the host is dead, then they can able to draw the nutrition for growth and multiplication, or this is we call it as a hemibiotroph. Necrotroph, right? So necrotroph is something different. So that pathogens, while entering, it will kill it. Right? First kill, then draw nutrition. Or that is from once the infection initiating time, so that the toxins produced by these organisms first kill it, then draw the nutrition. That is something different, right? For the hemibiotroph, initially it need for originate the infection, so initiate the infection, then it will kill at a later stage. But here necrotroph means first kill, then draw the nutrition. That is we call it as a necrotroph. So pathogenicity is we call it as an ability, relative capability of the pathogen that can cause the disease. We call it as a pathogenicity. So pathogenicity is measured through the genes, right? The virulence gene governed by the pathogens. That is we call it pathogenicity is an ability of the pathogen that can cause the disease. That is we call it as the pathogenicity. We call it as a courage, or sometimes in the case of pathogen, we call it as a virulent. It's a virulent. It's governed by the gene. It's a character. It's a character. So pathogenesis is a process, right? That is an ability. This is a process caused by the infectious agent. So process mean pathogen should come and contact and recognize. Then once the recognition is successful, then they will produce some certain chemicals and other things, then penetrate. That is the process we call it as a pathogenesis. Pathogen starting infections. That is we call it as a process. Right? Process we call it as a pathogenesis. Pathogenicity is an ability, right? So while the pathogenesis is going, the one state is a pathogenicity. While recognizing, if the pathogen is overwhelming the host, then it's a pathogenicity, right? It's because of this defense certain virulence factor produced by the gene that can able to dominate the host influence, right? Host. This is we call it as a pathogenesis. And virulence, degree of infectivity, that is measured through the pathogenicity. Relative capability, this is we call it as a degree of infectivity. While pathogen is infecting, so how much is a prediction, how much this or how extent this pathogen can cause the disease. That is we call it as a degree of infectivity of a given pathogen. That is we call it as a virulence. So there are important terms are there: barotrophs, necrotrophs, pathogenicity, pathogenesis, virulence. Right? These are related with the mainly pathogens. Another one is there an infection. It's an initiation and establishment of the parasite within a host. We call it as an infection. This is need not to be a pathogen. It may be anything, right? Any organisms. So initiation, establishment of a parasite. It's a broad term. Parasite, we call it as an infection. An infection. So if we this term explains that parasite. So this parasite mean we are not considering here the insects. We are considering here only microorganisms as well as some of the parasitic plants. Right? Why? So this is term we call it as an infection. Another term is the infestation is related with an animals. Infestation related with an animal. For example, if the insect is damaging plant, we call it as an infestation. Or monkey is damaging the plant, we call it as an infestation. The infection is mainly we are dealing with the microorganism or the pathogens and the parasitic plant. That is we call it as an infection. So primary infection means the first infection of the plant by overwintering or oversummering of the pathogen. This is I explained very well in the disease cycle. So there's a primary infection. Inoculum means any part of the pathogen that can cause the disease, we call it as an inoculum. Right? Portion of the pathogen which is transferred to the plant or cause the disease, or any part of the pathogen that can cause the disease, we call it as an inoculum. Invasion means penetration and spread of the pathogen. Penetration and spread, we call it as an invasion. Right? So colonization means growth and development. So that is, these all are from the infection to symptoms. These all are part of life cycle. So you have studied the life cycle. These from 9 to 15 are part of life cycle. Infection, so primary infection, inoculum, invasion, colonization, inoculum potential. So inoculum potential mean, so the growth or threshold of the fungus available for colonization of the substance. Mean that's it indicates that amount of inoculum. So 1 unit of inoculum we already discussed. 1 unit of inoculum we call it as a propagule. right? Inoculum is a part of the pathogen that can cause the disease. Inoculum potential means amount of inoculum present in a particular host surface. So the infection site, we call it as an inoculum potential. Right? Growth or threshold of the, we are mainly focusing here fungus, even bacteria also true, right? So, and symptom is an external or visual expression due to the pathogen attack. We call it as a symptom or alterations. External or internal reaction or alterations of plant as a result of the disease. We call it as a symptom. Right? Symptoms. Incubation period is a time period. Right? This is a period. Time period between from the penetration to appearance of the symptom. Penetration to appearance of the symptom is an incubation period. That's what I explained with the COVID-19, because from the infection, so until the expression of the symptoms of the fever and other things, it will take 14 days. That is an incubation period. That's what they are taking PCR in the first day and 14 days. It's an incubation period. Mainly many pathogens for incubation period, for especially the funguses in case of plants, so nearly 14 days, 7 to 14 days incubation period. For bacteria is 48 to 72 days, 72 hours you can see mainly. So disease cycle, we have studied the chain of events involved in the disease development. So there are two types of disease cycle, single disease cycle and multiple disease cycle, or monocyclic and polycyclic. Monocyclic mean, so the disease or the fungal or any bacterial or virus can complete the life cycle of the disease or life cycle of the pathogen only one time of the particular crop season. For example, if we are starting the cultivation of the paddy from the seed transplanting until harvest, the pathogen can able to complete the life cycle only one time. That is very coincided with the crop phenology. For example, if you are cultivating any BG1111, 3.5 month variety, so this macropotha disease, blast disease, so infecting on the rice. So this rice blast can complete a life cycle only one time for the whole complete 3.5 months. Right? That is, we call it as a monocyclic. So mainly these fungus are showing monocyclic. For example, downy mildew on rapeseed. So downy mildew is brassica, right? Brassica crops. So you can see the downy mildew of rape and clubroot of crucifer, Plasmodium brassicae, and sclerotia blight on brinjal, right? So these are completing only one cycle. And multiple cyclic mean within the between the one season, for example, in the rice, the same rice blast can complete the life cycle 3 to 4 times. For example, if you're taking rice blast, that is the example, the rice blast, blight, rust, right? So those pathogens can complete a life cycle more than one time of the one crop season. That's what we call it as a multiple cycle. Multiple cycles. So disease syndrome. So disease syndrome, the set of varying symptoms characterizing the disease are collectively called as syndrome. So syndrome, so mainly it's a collection of more than one symptom is present, we call it as a syndrome. Mainly the syndrome, the word in medically, they are telling that if this problem in genetic or disorder, they are calling it as a syndrome. Right? Syndrome. And alternate host. Alternate host mean other than the main host. For example, late blight of potato. We are calling it as a late blight of potato because it's initially identified in the potato, it causing the late blight in the Phytophthora infestans. These Phytophthora infestans, if there is no any bacteria, the potato in the field, if the farmer is cultivating the same family crop, tomato, so these Phytophthora can cause the diseases on tomato. See, tomato is an alternate host. Or sometimes we are calling it mainly the alternate host, mainly we are calling that host parasitic fungi, the plant not related to the main host, it needed to complete its life cycle we call it as an alternate host. This is a typical example of the rust in wheat, especially stem rust, right? So the wheat, so these wheat fungi cannot produce sexual spores if there is no any alternate host. We call it as a barberry. Barberry plant are the alternate host for the wheat rust, stem rust. So if the barberry plant is there only, they will produce sexual spores. So that's how in the wheat rust studies, if you are going there and if you're finding the wheat studies, so all the governments of many wheat growing countries, their target is to remove or completely eradicate that barberry plant from there, because these are weeds. Wheat, right? Not wheat. Weeds. Barberries are weeds. So wheat fungi using this one as an alternate host to complete a life cycle. So this life cycle, reproductive life cycle. It's a collateral host. That is, I earlier told that. Wild host of the same families of the pathogen is called as a collateral host. Right? For example, if the tomato nematode, right? So root node nematode is attacking on the tomato, cultivated tomato. Sometimes wild tomato act as a collateral host for this pathogen. Right? And predisposition. Predisposition means effect on environment. One or more environmental factors that makes plant vulnerable to attack the pathogens. That is, we call it as a predisposition. Predisposition. For example, for that COVID-19, if you are vulnerable, if you are having that lungs related diseases or diabetic diseases, so you are prone to have that viral COVID-19 attack more than other people. That is because of this factor, your host condition, right? So somebody, so if you are going there in the whole condition, they cannot able to tolerate. If somebody, they are going in the hot condition, they cannot able to tolerate. So because these environmental factors influence their body. Like that, some plant, the conditions are influencing and weaken the host resistance. Therefore, so this it's a time for the pathogen to multiply. That is, we call it as a predisposition, the effect of environmental factors. For example, in the high dew condition, these environmental conditions, the dew conditions or high dew conditions, the host will become bit wetty. So therefore, the more fungal pathogens can spores can then land and initiate infection. So at the time, so temperature will be low, then more host metabolic activity will be low, then they cannot able to take defense reaction immediately. So that is we call it as a predisposition. And physiologic race. At physiologic race mean, so some of the organisms, they have the similar morphological character, so, but their biochemical characters are something different. That is we call it as a race. For example, so if you are taking that fusarium, fusarium is a wilt-causing pathogen. So they are producing similar morphological structure. They are producing similar spores, but all the fusarium cannot attack in all the hosts. For example, fusarium oxysporum, former species, that's what we are mentioning, the former specialis, Lycopacisi. So Lycopacisi means Lycopassicum esculentum tomato. So Lycopacisi strain can attack on tomato. Fusarium oxysporum, former specialis cubensis, that is causing the banana wilt in banana. Cubensis, Musa cubensis. It's a plant, Musa baltesiana. It's a host plant, banana plant. So the Fusarium oxysporum forma specialis cubensis means that can able to cause the banana wilt on bananas. not in tomato and other things. That is we call it as a physiological race. Right? So in the even the oxysporum also, there are 4 races are there. Race 1, race 2, race 3, race 4. Even in the banana variety, some Fusarium oxysporum form species like cubensis, maybe race 1 can attack on amble, Simon Kadali. Race 2 can attack on colicoid, couple. Sometimes the race 1 and race 2 can attack on ash plant. Right? That is we call it as a physiological race. According to that, you will in the host type, you will divide into biotype 1, biotype 2, biotype 3, like that. Right? That's what we are developing the varieties. Different rice varieties we are developing. Why? For the to create the variation against resistant pathogens. One target. We will study. I will explain this more in the host pathogen genetics. Right? In the molecular term, how we can progress. Right? And the biotype. They are here. Another one is the biotype. Similar morphological unit within the species, the members of which are usually genetically identical. That is we call it as a genetically they are identical, but we are calling it as a biotypes because of the some of the biochemical reactions of that is we. Right? So that is okay. I told you so. The BG variety 250, 270. So genetically they are similar, but a single portion sometimes. So biochemical is it different? That's what we call it as a biotype. Another one is the symbiosis. Could have studied this one very nicely in the rhizobium. Right? Rhizobium. So beneficial association. 2 organisms are getting beneficials. So another term is symbiosis and mutualism is very close, right? So symbiosis, both organisms get benefits. For example, that is a very legumes with the rhizobium. The mutualism is symbiosis of 2 organisms that are mutually helpful or mutually support one another. That is also a similar term. That's what you see. Symbiosis of 2 organisms that are mutually helpful. You one give, another give, take. But mutually beneficial association, 2 or more different kind of organisms, they are living together because they need for the survival. So this is in this one we studied the rhizobium plus in leguminaceous crop. This mutualism, we study some of the crops with an vascular mycorrhiza. Mutualism. Antagonism means counter reaction of the organism or group of organisms. We call it as antagonism. It's a trichoderma. One organism will not allow the growth of the other organism. We call it as an antagonism. These terms are 3 terms are very important if you are studying in the biological control of beneficial organisms and the pathogen. Symbiosis, mutualism, antagonism, and parasite. There are 4 terms will be there. Parasite, symbiosis, mutualism, antagonism. Antagonism is beneficial effect of organism to protect the plant from the pathogen. It's antagonism. Against pathogen, trichoderma giving beneficial potential. For symbiosis, the host is giving room for the growth of the pathogen, so both are get benefit. This mutualism means photosynthetic product is given, so the at a part of the host, these microorganisms are working. That's mycorrhiza. Right? Parasitic means the host, there is no any beneficiary. Here, one is getting at least benefit. There, so only pathogen is getting benefit. Negative organism, unwanted organism get benefit. Here, the wanted organisms get benefits. So unwanted organism get benefit. We call it as a parasite. Mutation. Mutation, you all know, so sudden, abrupt changes in the genetic material or the character. Sudden changes in the gene, genetic material. We call it as a mutation. So mutation is a reason for we creating variations. One of the variation in pathogens. So likewise in the COVID-19, there are different strains. So in the now it's a very big problem, right? So they have produced a vaccine. Even in the Britons, they have identified there are 4 strains due to mutations. One of the strain is related to the South African strain. The South African strain will not work against any vaccine, whatever the vaccine so far produced. It's not working. So virus is mutated. That's what the British government banned 33 countries. For South African, the African countries, people, and some of the virus spread into this African strain lineage. We call it as a lineage. African lineage spread countries, people cannot enter the Britain 33 days. So from yesterday they announced 33 countries, people cannot enter it. Even sometimes in Sri Lanka, we don't know because whether we have that, because we are taking people from different part of the country. If people are coming from with this strain, then even Britain also put a ban to Sri Lanka to not come. And also in end report, everywhere they are putting in the quarantine now. So because it's not under control in British and USA, people every day, 2,000 people are dying. So this is because of mutation. Pathogen virus is a sudden mutation. It can cause sudden mutations. That's the problem. So if you are developing vaccine for the viruses, it's very difficult. It's true for even plant viruses. So we cannot find any viruses. And disease. Disease is a normal, not normal. If an abnormal, we call it as a disease. Deviation from the general health or physiology or function of the plant, we call it as a disease. If it is a severe disease, then the plant or the host will deceased. Disease, D-I-C-E-A-S, disease means death. D-I-S-E-A-S, disease means not normal. If it's a disease, severe condition, you will deceased, die. Right? So be careful with the spelling. And crop damage. So crop damage, or not is mentioned as a crop damage. Please change it there. It's a wrong crop damage. The crop damage is the right. Crop damage is the any reduction of the quality and the quantity of the yield or the profit or revenue from the crop due to the pathogen or attack, we call it as a crop damage. Yield, mainly we are telling the yield losses, quality losses, both. Yield is a quantity losses and quality losses. Or management cost is high, we call it as if it is a damage. This is due to the sudden reduction in quality and yield of due to the crop injury caused by the pathogen. Or any organism, even insect also we are mentioning the crop damage. Deficiency means abnormality of the particular nutrient to the crop or suboptimal level. We call it as a deficiency. Essential nutrient is not there. The plant show the symptom. These symptoms, if you are misinterpreted as a pathogen attack, definitely you will spray the chemicals, unwanted chemicals, but actually it needs nutrition. This is mainly creating the nematode attack, normally creating the confusions to the nutrient mimic. Because the plant show the yellowing symptoms and dropping of flowers and fruit. So the farmers are confused. This is because of nitrogen and other deficiency. They are applying more amount of nitrogen. It creates more diseases. Right? So therefore, the proper diagnosis is important, and we will discuss later in the proper diagnosis. So deficiency is an abnormality of the or disease due to the lack of nutrition. We call it as a deficiency. It's a symptom, not a syndrome. It's a not the symptom. It's a syndrome. Deficiency syndrome. Not deficiency symptoms. It's related, but mainly we are calling it as a deficiency syndrome. Right? Pathogen disease symptom. Insect infestation. So please don't forget it. Insect infestation or the high animal infestation, microorganism infection, nutrition deficiency is a syndrome. Symptom is you are very clear. Syndrome you are very clear. Pathogen is you are very clear. Inoculum and everything. There are 32 terms. If you are very clear with these 32 terms, you can answer if anybody asks a question anywhere. Not only for the exam point of view, anywhere of the place. Right? That is important. So basic understanding. So until now, if you have any questions, so please you can ask questions. Mode of attack of plant by plant pathogens. Right? This is an important task. So we have to deal with why. So how that plant pathogen attack its host? What is the way they are using? What are the chemicals or weapons they are using? Sometimes we call it as a weapons of plant pathogens. right? Weapons of plant pathogen. So we will go from the outside to inside, right? So first contact the surface, right? For the contact portion of the plant is the host surfaces, right? Host surfaces. So the aerial plant surfaces mainly consist of cuticle, right? So in the epidermis, so you can find it the cuticle. So in the root surfaces, they consist other than the cuticle, they are consisting celluloses, mainly celluloses. So some plant surfaces you can, instead of cuticle, they say some cuticle are very thicker cuticle, some cuticles only having the cellulose only. So other things, pectin, no wax layer, other things you cannot find it, right? So cellulose primarily they are consisting of the cuticle mainly consists of cuticle, right? So lower cuticles, so lower epidermis layers mainly predominantly consist of pectic substances. These pectic substances and hemipectins, so pectin and the hemipectin and polysaccharides or lignin all together to form a cell cuticle and epidermis and cell wall of the plants. If the lignin is present, it's very hard. If cutin is present, it's very hard. Once it's suberonized, it will further harden, right? So if you want, this is a first defense zone of the plant, right? So like a skin of your body, it's a first defense zone. If an enemy want to create the trouble to the host, the area of nucleus, is main part or central point, the pathogens have to break all these things. It's a defense zone. It's a first defense area. So in the first defense zone, so the pathogen should break it. In the first defense zone, other than the cuticle and cell wall, we are looking at that as a physical structure. So if you are going there in the electron microscopic picture, other than they have the radar system, like that radar system I am saying. So like an communication system. Plant cells, all the cells having that communication system. This is we call it as a like a radar system to know the environment, immediate environment. It's like a radar system, right? Radar system. So because it's a defense area, no? The plant's defense area, it's like a battle. We are dealing with like a battle. If the pathogen is coming and contact, so until if the pathogen is producing any chemical substances or is trying to use any weapons, physical weapons, stylet or other things, so this area, I mean defense system, air defense like system, will not recognize, the plant defense system will not recognize anything, right? Well, this is a cell wall complex, see, it's a complex, will not recognize anything. So therefore, this system should be the first area the pathogen will target to break, right? So this is a composition of the cell wall, right? The cell wall composition, how it is made it is a the electron microscopic picture, right? So middle lamella and primary and secondary cell wall all together to form a cell wall. And the middle lamella you can see is bipectinized. Pectin and semipectins are there. So in between cells, so there is a middle lamella is hardened, right, by using the hemicelluloses. So in the cell wall structure, so mainly they are having the certain components, right? They are proline-rich proteins, glycine-rich protein, solanaceous lectins, and albinogalactan proteins. These are the certain components will be there. These components will be also need to break by the pathogens. Here we are dealing with mainly fungi. So because the fungi are using the mechanical structures or the chemicals directly create the injuries. So bacteria and viruses, they will penetrate by the injury created by the other organism or naturally. That's what here everywhere we are taking the fungi, right? So nematode is a different section, but nematode also is using the same technique, but nematology we are in the specialization area, right? For if you are planning to have the agriculture biology specialization, you will study on that, but we will touch certain nematode here, right? So this accumulate in response to elicitor. So once the pathogen is coming and contacted, so pathogen will produce chemicals. I told you. These chemical molecules are elicitor, right? The elicitor molecules received by these people. I told you, these are the defense system, right? Radar system like that. These protein will receive these elicitor molecules, mean pathogen produce chemicals. So then the message will be passed to the nucleus by this thing, this network. Then the nucleus will order to the surface to trigger the defense system, in which way the triggers the defense system should work, maybe physically or chemically, right? So that's how these certain components will be there. Then the mode of attack of the plant pathogen, they are using there two ways. One is a physical forces, other one is chemical weapons. Against to defend, host also produces, there are two things: mechanical barrier or physical barriers, and chemical weapons. They are also using the chemical weapons and mechanical barrier. So we will first study the pathogens weapons, mechanical barrier, mechanical defense forces, and the pathogen weapons. Then we will study that plant defense, right? Plant defense. So pathogen, so they are using the mechanical forces. These mechanical forces can be created by these structures and in with the facilitation of these enzymes and of the certain tools the pathogens have. First, once the pathogen host post is attached to the host surfaces, immediately, if it is a fungi, they will produce a germ tube. Right? Germ tube, while growing, definitely at the end of the germ tube, they will produce a structure we call it as an aprosorium. This aprosorium can produce lot of amount of enzyme. That enzymes may be cellulases, chemipectinases, pectinases, lignases, cellulases. So these enzymes, all enzymes can able to degrade the cell wall and epidermis component of the plants. Right? So the aprosorium will eject all these enzymes around this plant cell wall. So adhesion of the pathogen. So an aprosorium produces chemicals. Once this chemical is disintegrated that area, then end of the germ tube, very closer to the aprosorium, another structure will be there. I told you it's a very narrow, delicate mycelia. We call it as a penetration peck. This peck, just like a nail, a needle, just make a push. If it is make a push, the area is collapsed. If it is a push, then quickly this mycelium can penetrate. Like an enzyme, nematode. So nematode will do first in different way. First, they will make a puncture through these stylet needles because they have the mechanical forces. Their organism can move independently, penetrate. When the stylet is penetrate the host tissues, then through the salivary gland, they will eject these enzymes into the cell directly. Right? So that is the mechanical weapons. This is we call it as a mechanical way how the pathogens break the host. Right? Break the host. This is a example pictures, right? I told you, suppose here germ tube, here under the germ tube, this is produce aprosorium, this is produce lot of enzyme. So this Aprosorium eject the enzyme here, then penetration peck suddenly piercing. So nematode, they have the stylet, a slight stylet, make puncture, then eject enzymes. So these chemical weapons are different categories. There are physical weapons, these are chemical weapons. So these chemical weapons, we will study under the 5 different categories. They may be enzyme or maybe toxins, maybe growth regulators and polysaccharides. 4 categories: enzymes, toxin, growth regulators, and polysaccharides. Right? So once the mechanical forces created the initial damage, then there is no any mechanical forces used by the pathogen. Only the chemical forces will be used. Chemical weapons will be used by the pathogen. Right? So this chemical weapons can important to decide the pathogenicity of the particular pathogens. Right? So pathogenicity. If the enzymes is overwhelmed by the defense, then the pathogenicity is less. These toxins are very potential and very toxic, can able to overwhelm the defense of the host. So pathogenicity is very high. It's a high virulent pathogens, right? High virulent pathogens. So these are 4 types of enzyme. So enzyme role is to disintegrate. Enzyme role is to disintegrate the component of the host cell and break down of the food and food substances into absorbable forms and change the membranes and protoplasmic compositions and its functions directly. These are the role of enzymes. And toxin, how it works. Toxin, they are changing the protoplast components. In the protoplast, there are certain components will be there. These components will be handle the membrane permeability, other stomatal closure and other things. But if it is toxins interfered, this system will get collapsed. System by system, they are collapsing, right? First, this cell membrane permeability is collapsing, protoplasm membrane is collapsing, and growth regulators. The growth regulators is copious amount is released into the plant cells. So this hormones will induce the function of the certain things or reduces the function. Right? If it is an induces the function, cell will multiply and enlarge it. Plant will grow very fast. If it is, or sometimes the growth will be retarded, but proliferate more amount of branches and leaves. The growth regulators is reduced, then the plant will be very short, less produced. And polysaccharides. These polysaccharides are produced massively into the xylem tissues and phloem tissues. That's how the food translocation, water translocation is interfered. Then your plant will show the will symptom, not symptoms. So not all the pathogen will not use all the chemical weapons. It's depend on different pathogens. Some pathogens can use mainly nematode, they are using the growth regulators. The enzymes is consisting mainly growth regulators like that. But wilt causing bacteria and funguses, especially wilt causing bacteria, they are using mainly polysaccharides. Many the fungal pathogens, they are producing enzymes. So these all the pathogens will not use all the weapons, right? It's depend on their virulence. Right? The enzyme, how it works. Enzymes. So enzymes, you all know the protein molecules, right? That catalyze all the interrelated reaction in the living cells, right? So these enzymes in the disintegration, what they are doing is, because sclerotina sclerotium, he studied this person. So it is producing certain enzymes, mainly the cellulases. These cellulases can work on in the collar region of the plant. If they are working on the collar region of the plant, that area will be disintegrated. So that's how your plant will get rot in that collar region. That is we call it as a collar rot. Immediately after the rainy season, your many plants will get wilt and die immediately. So because you don't know the reason, this is because of the collar rot. Right? Collar rot. Even damping of pathogen. So fungal, the Pythium fungi, Pythium aphanidabatum, Pythium dephrianum, and Phytophthora, they are also causing the damping offs. This damping of these spores also, they are producing enzyme. So immediately after the emergence of the seedling, before emergence of the seedlings from the seed, the cotyledon region is working in the cotyledon region or the plant aerial root junction. That's what your seedlings will get collapsed. The area get rot. There is no transfer because of these enzymes. So these enzymes are maybe proteinases, pectinases, and cellulases. Right? So we will study the one by one. Cutinases and cellulases and lignases often secreted by these plant pathogenic functions. And nematode and fungi, they are also produce more. Their target is to further disintegrate these cell wall and epidermal cells, epidermal region and cell wall component and cell membrane of these plants. The second category is the microbial toxins. There are two different type of microbial toxins are there. One is host specific microbial toxin. Other one is host non-specific microbial toxins. Right? So we will study next class and we will continue the rest of these chemical weapons of the plant pathogen. So until now, you have any